Welcome to this lecture on structural bioinformatics. Today, we are going to explore one of the most fascinating areas of modern science. We will learn how computers help us visualize and understand the tiny, invisible three-dimensional machines that keep our bodies alive. By the end of this session, you will understand how the physical shape of a molecule dictates its biological function, and how we can mathematically measure these shapes to design life-saving drugs. Let us dive right in. To begin, imagine you are trying to understand how a complex car engine works. If I just hand you a written list of parts, like screws, pistons, and belts, you would have almost no idea what the engine actually does. However, if I show you the fully assembled three-dimensional engine, you can immediately see how the pistons pump. In biology, a protein's linear sequence of amino acids is just that simple parts list, but its folded three-dimensional shape is the fully assembled working engine. Structural bioinformatics is the exciting science of using computers to understand these incredible biological machines. Now that we understand why the three-dimensional shape is so important, let us formally define our topic. Structural bioinformatics is a highly specialized subdiscipline of computational biology. Instead of just looking at the linear sequence of genetic letters, this field uses powerful computational tools to analyze the exact spatial coordinates of every single atom in a molecule. Researchers focus primarily on biological macromolecules, such as proteins and nucleic acids like DNA and RNA. This field also involves storing these massive three-dimensional structures in global databases, like the Protein Data Bank, and visualizing them on computer screens. Building on that definition, structural bioinformatics does much more than just store known structures. It allows us to predict the shapes of entirely unknown proteins using advanced algorithms. Ultimately, this field bridges the massive gap between a simple, one-dimensional genetic sequence and the complex physical reality of how molecules interact in three-dimensional space. It is truly the ultimate intersection of biology, physics, and computer science, bringing together biological principles, physical forces, and computational power to solve complex biological puzzles. This brings us to an important question. Why do we care so much about the three-dimensional shape of a protein? The most fundamental rule of molecular biology is that structure determines function. The physical shape of a molecule dictates exactly what it can do. For example, the enzymes in your stomach have a specific three-dimensional pocket that perfectly fits the food molecules you eat, acting exactly like a lock and a key. If a genetic mutation changes that three-dimensional shape even slightly, the key might no longer fit, which can lead to severe genetic diseases. Understanding this lock and key mechanism leads us directly to real-world applications. Pharmaceutical companies use structural bioinformatics every single day to design new life-saving medications. By knowing the exact shape of a disease-causing protein, scientists can use computers to design drug molecules that perfectly fit into the protein's three-dimensional pockets. Once the drug binds to the pocket, it effectively shuts the harmful protein down, curing the patient. This rational, structure-based drug design is a massive leap forward from traditional trial-and-error methods of discovering medicines. Now that we see how crucial these shapes are for drug design, we need a way to analyze them quantitatively. When we use computers to study these three-dimensional structures, we often need to compare two proteins to see exactly how similar their shapes are. To do this objectively, we cannot just rely on visual inspection. We use a mathematical measurement called the root mean square deviation, or RMSD. This metric calculates the average physical distance between the corresponding atoms of two superimposed proteins. Understanding RMSD is absolutely essential for anyone working in computational biology. This concept of measuring similarity brings us to the RMSD formula itself. Conceptually, this formula finds the distance between matching atoms, squares it to remove negative signs, averages those values, and takes the square root. Let us look at the symbols. N represents the total number of atoms we are comparing. The variables x1i, y1i, and z1i are the three-dimensional spatial coordinates of a specific atom in the first protein. The variables x2i, y2i, and z2i are the coordinates of the matching atom in the second protein. If the final RMSD value is very small, close to 0, the structures are nearly identical. As the RMSD value increases, the structures are physically more different. Before we move on, let us see this formula in action with a concrete example. Imagine we have a tiny, simplified protein with only 2 atoms. We want to compare its natural structure to a computer prediction. For the first atom, both structures are at the origin, 0, 0, 0. Subtracting these coordinates gives a squared distance of 0, meaning perfect prediction. For the second atom, the y-coordinates differ by 1 unit, from 3 to 4. Squaring -1 gives a positive squared distance of 1. We add these squared distances, 0 plus 1, and divide by our total number of atoms, which is 2, giving 0.5. Finally, taking the square root gives approximately 0.707. This means the predicted atoms deviate from the natural structure by about 0.7 units of distance. Now that we have used this formula on a simple protein, let us apply it to a real-world drug docking scenario. A researcher knows a drug atom must bind at coordinates 5, 5, 5 to cure a disease. A computer predicts it will bind at 8, 9, 5. We need an RMSD under 2.0 for success. Since we track one atom, n equals 1. We subtract the coordinates. 5 minus 8 is -3, 5 minus 9 is -4, and the z-coordinates cancel out to 0. Squaring these gives 9 and 16, which add up beautifully to 25. The square root of 25 is exactly 5.0. Because 5.0 is much larger than our acceptable threshold of 2.0, we conclude the computer prediction is poor, and the simulation must be run again. To truly grasp what we just calculated, let us look at this visual representation of superimposed protein structures. On the screen, you see 2 folded protein ribbons. The blue ribbon represents the true natural structure of the protein, while the red ribbon represents the computer's predicted structure. Notice how they follow a similar overall shape, but do not align perfectly. The dashed line between them highlights the physical distance between corresponding parts of the molecule. When we calculate the RMSD, we are essentially measuring the average length of all these dashed lines across the entire protein. This diagram perfectly illustrates why structural bioinformatics is so powerful. It allows us to visually and mathematically quantify the 3D machinery of life. We have covered a lot of ground today, so let us summarize the most important ideas. First, remember that structural bioinformatics is all about analyzing the exact 3-dimensional spatial coordinates of molecules, moving far beyond simple linear genetic sequences. Second, the golden rule of molecular biology is that structure determines function. The physical shape of a protein dictates exactly how it behaves in the human body. Finally, we learned that the root mean square deviation, or RMSD, is the essential mathematical tool used to objectively compare these shapes. By mastering these concepts, you now have a strong foundation to understand modern drug discovery and the physical basis of molecular biology. Great job today.